Okay, so so far the embryo have been developing inside the zona pellucida. Now the zona pellucida, remember, is a protein matrix that is protecting the embryo. So the embryo did not bind to the isthmus during the travel from the ampulla to the isthmus to the uterus. This is extremely important because it avoids extrauterine implantation and ectopic, which result in ectopic pregnancy. And ectopic pregnancy are very dangerous for both the fetus and obviously the mother in all mammals. And obviously, we are mammals in our species, but other species too. However, at this point, the zona pellucida become an impediment. So when the embryo reach or is reaching the uterus, you need to get rid of the zona pellucida. And how does it do that? Well, the trophoblast, remember, the group of cells that are surrounding the blastocyst, and they are basically external in the embryo. And in this nice picture here, you see the trophoblast all around. And then in the middle there, this thickening is basically the ICM. So the trophoblast produce proteases. Proteases are obviously enzymes that digest and break down proteins. And these proteases create a hole in the zona pellucida, and the embryo is able to hatch from that hole and exiting the zona pellucida and be available to be to bind the uterus. Now, the uterus in the meantime have been undergo a number of changes. The endometrius, which is the uterus epithelia, because of the action of different hormones, including estrogen and progesterone, produce an extracellular matrix of proteins that are there to catch the blastocyst. And here you see in this nice picture the endometrium of the uterus that coat the embryo. You can recognize all around the trophoblast, and the ICM is that thickening in the middle. Okay, so at this point, the embryo wants to do one thing and one thing only. The embryo wants to bury itself inside the maternal tissue, so inside the uterus. To do that, the trophoblast produce a set of proteins that are called integrins that digest the uterus epithelium and allow the embryo to enter the uterus walls. You can see here the embryo that is trying to invade the uterus epithelium and then move as much as possible inside the uterine tissue. So the blastocyst is able to enter or try to enter the uterine wall. And of course, the embryo at this point doesn't have much nutrient available to it. So remember that in mammals, we don't have rich yolk proteins. We don't have a lot of yolk protein available to sustain the growth of the embryo, like it happens, for example, in birds and amphibians and reptiles. In mammals, on the other hand, the mother undergo a transformation to produce some tissue that sustain the development of the embryo. So everything actually starts from the trophoblast. The trophoblast, which is obviously part of the embryo at this point, the trophoblast develop into the chorion. The chorion is the embryonically derived part of the placenta. The trophoblast also induce the cell of the uterus, so the maternal uterine cells, to produce the decidua. And the decidua is the maternal part of the placenta. So the placenta, which then sustain the development of the embryo, as a part that is derived from the embryo itself, from the trophoblast, which is the chorion, and another part that derived from the maternal tissue, so the uterine tissue, which is the decidua. So what's happened now? What's happened at this point to the mammalian embryo? So these days here refer to human's embryo, but remember that we are not simply talking about human embryos here, and we are talking about processes that we can find and they are common in most mammals. But obviously other mammals do not have this development done in the same number of days. It can be shorter or longer, depending on the mammals. So what's happened to the embryo now at this point, and what different part of the embryo are going to do in terms of the development of different part of the fetus. So we have our blastocyst, and in the blastocyst we recognize the trophoblast and the ICM. The trophoblast is going to produce two major tissues, the cytotrophoblast and the syncytiotrophoblast. Now, here you see the embryo that is trying to go inside the uterine tissue, and here is the same embryo a bit later when it's already on its way inside the uterine tissue. You can recognize this structure here. This structure is derived from the trophoblast cells that are growing very fast inside the uterine walls and producing the chorion. The chorion is basically the syncytiotrophoblast. Excuse me. The trophoblast is also growing, forming an extra layer of cells around the embryo, while the embryo is moving inside the uterine tissue. The syncytiotrophoblast also try to expand and make contact with the blood vessel of the mother and inducing the formation of the decidua as well. The ICM, on the other hand, is divided in the hypoblast of primitive endoderm and the epiblast. So if you look here, if you look on this sketch or this cartoon of the embryo, you see that we have a green line of cells. This green line of cells is represented by the hypoblast. Are the cells of the ICM that are in contact with the blastocoel. These hypoblast cells, or primitive endodermal cells, are going to form the yolk sac. Okay, so they're not really going to form any tissue of the future embryo. The epiblast cells, on the other hand, are the cells that are going to form all the embryonic tissues, the endoderm, the mesoderm, and the ectoderm. The epiblast is also going to form the amniotic tissue, the amniotic cavity. Okay, so here we have a later embryo. In this later embryo, we can recognize a syncytiotrophoblast that grows and connected with the blood vessel of the mother, forming the chorion. We have the cytotrophoblast that form a thickening around a developing embryo. In green here, you can see that the hypoblast grow all around the blastocoel or the blastocystis cavity, forming the yolk sac, while the epiblast form an initial amniotic cavity that is going to be filled with amniotic fluid, and this in blue represent the amniotic ectoderm, which derive directly from the epiblast. Now, the layered cells here in orangey above the hypoblast are the only cells that are going to form the embryonic ectoderm, the embryonic mesoderm, and endoderm. So they're the only cells that are going to form the fetus. Okay. At this point, we conclude cleavage, and now we can talk about gastrulation. Now, our embryo, the human embryo, as other mammalian embryo, at this point undergo gastrulation as in exactly the same way as you can see done in birds or reptiles, and even amphibian. Why is that? Well, evolutionarily speaking, our embryo remember the common ancestor with this other group of animals. So it is quite probable that the ancestors of all these group of animals, different animals, birds, mammalians, amphibian, and reptiles, were laying eggs. So our embryo developed, or the mammalian embryo, developed as if there was a huge yolk full of yolk protein just below them. So it developed as it is on top of a gigantic yolk, like the embryo of birds or in lizards that develop on top of a large yolk. So gastrulation is reminiscent of this other species. And this is one of the reasons why we use a lot chicken embryo in developmental biology to study this, in particular, this stage of development. Because understanding how this stage of development works in chicken embryo allows us to understand how it works in other animals like us and other mammals, because the stages are basically identical. So what we see when we look at the embryo at this point of development, so the mammalian embryo, which is actually very similar to what we see in other vertebrates as well. So we see what is called a bilaminar germ disk. In this bilaminar germ disk, we recognize two layers of cells. One layer, which is the epiblast, and one layer, which is the hypoblast. Yes, the hypoblast grow all around the blastoquel of the blastocystis cavity to form the yolk sac. Now, this is happening also in a chicken embryo, for instance. However, the yolk sac that is forming in the chicken embryo will include the yolk, which is deposited there by the mother. But here we don't have this yolk. We just have a cavity that is full mostly of water and some salt. So, however, the embryo developed as if it was on top of this massive yolk, and it formed even a yolk sac that theoretically should contain the yolk proteins, even if they're not there. When this yolk sac is formed, we can recognize two kinds of hypoblast. We have the visceral endoderm, which is the one that is near the epiblast, and we have the parietal endoderm, which is the one that is far away from the epiblast and the one that basically forms the yolk sac. The epiblast itself has formed an amniotic cavity, which is surrounded by epiblast cells that are forming the amniotic ectoderm, and this amniotic cavity is full of, or is going to be full of amniotic fluid. Okay. At this point, something very important is going to happen in the embryo. What is important in this nice cartoon, we can see our embryo from above. We can see our embryo from above, and we see here in gray the epiblast. In green, we have the hypoblast. Below, there is the yolk sac. On top, on blue, there is the amniotic cavity. And we see here the formation of what is called the primitive groove and the node. The node is a thickening on the epiblast. It has a funnel shape, and the cells of the epiblast can move through the node downwards and in an anterior direction. The cells can also move through the primitive groove, and they're moving downwards and laterally. The node is initially formed posteriorly, and then it moves toward the anterior part of the embryo. We can look at a section. So if we cut a section on this level, where you can see this dotted line, we are going to see the embryo like in these pictures. So we recognize our node. We recognize our node and the cells of the epiblast. The cell of the epiblast migrate through the primitive groove, and the first cell that migrate through the primitive groove are going deep and substitute the primitive endoderm or hypoblast, forming the actual germ layer, the endoderm. So the endoderm is constituted by the first cells that move through the primitive groove. They go deep and remove the epiblast remove the primitive endodermal cells, the hypoblast, remove them, substitute them, and form the real endodermal cells or the real endodermal layer. The second group of cells that move there are going to stay in the middle, forming the mesodermal layer, the mesoderm. The rest of the epiblast cells are actually staying outside, and they are going to grow all around the embryo, forming the epiblast. Some of the cells move through the node. Remember, the node initially is formed posteriorly and then it starts to move anteriorly. Some of the cells that move through the node and move anteriorly are going to form a tubular structure that is called the notochord. And the notochord is very important because it induces the formation of the neural tube, so the nervous system. Here in these nice pictures, nice cartoons, we have a representation of a chicken embryo. And we are seeing this chicken embryo from above. And at this stage of development, the development of the chicken embryo is almost identical to the development of a mammalian embryo and a human embryo. So you can see here the formation of a primitive streak or primitive groove that initially is formed posteriorly and then start to move anteriorly. You can recognize the node, which in the chicken embryo is called the Ensen node, just because this person Ensen described it initially in chicken. The cells of the epiblast are migrating internally through the primitive groove and through the node. Through the primitive groove, the cells are migrating internally and laterally. Through the primitive node or through the node, the cells are migrating internally and anteriorly. However, one interesting thing to remember is that the node initially originated posteriorly and move anteriorly, but at one point it starts to regress from the anterior to a posterior position. So the node initially moves anteriorly, and then when you reach the most anterior extension, it starts to regress posteriorly. As you can see here in these sketches, how the node initially moves anteriorly and then start to regress posteriorly. And this is very important to remember because while the node regresses posteriorly, the notochord is produced. So here you see in this graphical representation, you see the node that have moved anteriorly, and then when it is at its maximum expansion anteriorly, some of the cells that migrate through the node move anteriorly, forming the notochord, which is represented by these red lines here, and then the cells are moving, they tend to move anteriorly to form the notochord. However, at this point, the node is starting to regressing. So while the node is regressing posteriorly, the cells that migrate there are still forming the notochord, and therefore the notochord is formed from an anterior to posterior direction. So initially, the cells of the notochord are moving anteriorly. They keep doing so, but the node is regressing posteriorly, and therefore the node is for both in an anterior direction, but mostly in a posterior direction because of the regression of the node. In this developing embryo, we recognize also an anterior to posterior gradient of different signals that basically create a grid for the cells a grid that the cell can read to understand where they are along the anterior posterior axis. In fact, we have that the anterior cells, so the cells in the anterior positions, tend to express retinoic acid, while the cells posteriorly express FGF8 or FGF in general signals. And FGF create a posterior to anterior gradient, while the retinoic acid form an anterior to posterior gradient. Moreover, the retinoic acid inhibit the expression of FGF8, as well as FGF8 indirectly inhibit the expression of retinoic acid. So these two genes do not like each other. These two proteins do not like each other, and they inhibit each other expression. So while the notochord now is formed, the ectodermal precursor migrate to form the external layer of cells that represent the ectoderm. So at this point, we have already formed which is the end of gastrulation. We have formed the external layer, external embryonic layer, the ectoderm, the middle embryonic layer, the mesoderm, and the internal embryonic layer, the endoderm. Okay. So the objective of this lesson is to recall and be able to explain the different process of cleavage and gastrulation, and explain also the molecular and genetic mechanism that define the cleavage and gastrulation.